Hello, my name is Jones, and today we are going to look at the HIV life cycle. Some of the terms that we are going to use in this discussion include CD4 receptor, co-receptor, and T lymphocytes. When we say CD4 receptor, we are talking of the protein present on the outside of an infection-fighting white blood cells. CD4 receptors allow HIV to bind to and enter cells. Co-receptors. In addition to binding a CD4 receptor, HIV must also bind either a CCR5 or CXCR4 co-receptor protein to get into a cell. T lymphocyte. This is a type of white blood cell that detects and fights foreign invaders of the body. So these are some of the terms that we are going to use within this discussion. Let us now begin our lesson and talk about the stages of HIV life cycle. So stage 1 is binding and fusion. HIV begins its life cycle when it binds to a CD4 receptor and one of two co-receptors on the surface of a CD4 T lymphocyte. The virus then fuses with the host cell. After fusion, the virus releases RNA, its genetic material, into the host cell. So this is what is happening in the binding and fusion. So the HIV virus binds to CD4 receptor and the co-receptor. Then fusion, the HIV RNA is released into the host cell. Okay, so now let us look at reverse transcription. So an HIV enzyme called reverse transcriptase converts the single-stranded HIV RNA to double-stranded HIV DNA. This is done with the help of the enzyme reverse transcriptase. So when you're talking of reverse transcription, the enzyme reverse transcriptase play a major role. As the RNA of the HIV is combined to the human DNA, so you're going to have a double-stranded HIV DNA. Then stage 3 is integration. The newly formed HIV DNA enters the host cell's nucleus, where an HIV enzyme called integrase hides the HIV DNA within the host cell's own DNA. So the integrated HIV DNA is called provirus. The provirus may remain inactive for several years, producing few or no new copies of HIV. So in integration of this stage for HIV, the enzyme integrase plays a major role. And we are saying the newly formed HIV DNA, it enters the host cell's nucleus. So within the nucleus where DNA is formed, we're having an HIV enzyme called integrase that is now going to hide the HIV DNA within the host's own DNA. So now we are looking at the integration of the HIV DNA with the host DNA. So the integrated HIV DNA is what we call a provirus. Okay, so the host DNA has now been combined with the HIV DNA within the nucleus. So integration here is happening in the nucleus. That is a very important point to note. Okay, so integration, we have the enzyme integrase, and it is happening in the nucleus as the HIV DNA is now combined or integrated with the host cell's DNA. Let us now look at transcription. When the host cell receives a signal to become active, the provirus uses a host enzyme called RNA polymerase to create copies of the HIV genomic material, as well as shorter strands of RNA called messenger RNA. The messenger RNA is used as a blueprint to make long chains of HIV proteins. So in transcription, we are saying the host cell receives a signal to become active, okay, so that proviruses are produced. Or we are looking at the proteins that are needed for a new virus to be formed are produced. So here we are saying the provirus uses a host enzyme called RNA polymerase. So when it is using the host RNA polymerase, it is going to create copies of HIV gene type material or HIV genomic material, okay, as well as shorter strands of the RNA, okay, shorter strands of RNA, which we call messenger RNA. Okay, so this messenger RNA is the one that is now going to be used as blueprints to make long chains of HIV proteins. Stage 5 is assembly. An HIV enzyme called protease cuts the long chains of HIV proteins into smaller individual proteins. As the smaller HIV proteins come together with copies of HIV's RNA genetic material, a new virus particle is assembled. So what we are saying, after the transcription of genomic material of the HIV has taken place within the transcription, stage 4, these they will assemble, okay, and an HIV enzyme called a protease cuts the long chains of HIV proteins into smaller individual proteins. Of course, these smaller proteins are going to come together, okay, with the copies of the HIV RNA, so that they form a new virus, okay, or new virus particle can be assembled. So they pick up all those parts that are needed for the new virus to be formed. So once this has happened, we move on to that is stage 6. Stage 6, budding, okay, as budding. So budding, the newly assembled virus pushes out, okay. So when it pushes out, a bud is formed, okay. Bud, more like an inflammatory thing to show that now the virus is coming out. So bud is formed from the host cell. During budding, the new virus steals part of the cell's outer envelope. This envelope, which acts as a covering, is composed of protein and the sugar combination called HIV glycoproteins. So these HIV glycoproteins are necessary for the virus to bind a CD4 co-receptor so that it can infect other cells. So the new copies of HIV can now move on to infect other cells. Alright, so in budding, we are looking at the coming out now of the new virus from the host cell. So a lot of them will come destroying this CD4 cell. Hence, that's why we see in HIV the CD4 cells are monitored. And the reason we monitor CD4 count is because those cells are destroyed. Okay? And when the virus moves out from this cell, it goes to infect other CD4 cells, further reducing the number of CD4 cells, okay, in the body. Alright, so we have been looking at the HIV life cycle, and in the HIV life cycle, we have talked about 6 stages. So there's binding and fusion. Okay, if you want to talk about 7 stages, you can put binding alone and talk about HIV binding to CD4 receptors and the core receptors. And then when we want to separate this and talk of fusion, we are looking at the RNA of the HIV being released into the host cell. Then when you talk of reverse transcriptase, we are looking at transcription as a stage. We are just looking at the activities of the reverse transcriptase enzyme, okay, from HIV. Okay, that helps to form the HIV RNA to double-stranded HIV DNA. Okay, then after the reverse transcription, where the key enzyme is reverse transcripted from HIV, and the conversion, okay, of the single-stranded HIV RNA to double-stranded HIV DNA, we have 3 integration. In integration, we are necessarily looking at the HIV DNA being integrated into the host DNA. Okay, so when these have been integrated, we call that a provirus. And this provirus may remain inactive for several years, producing few or no new copies. Okay, so now after some time, then we have the transcription. So when the host cell receives a receives a signal to become active, this provirus is going to use the host enzyme called RNA polymerase to create these copies of the HIV genomic material, as well as a shorter strand of RNA, which we call messenger RNA. So it is this messenger RNA that is used as a blueprint to make long chains of HIV. So once these proteins have been formed, they will assemble somewhere. So stage 5 is assembly, where the where an HIV enzyme called a protease is going to cut the long chains of HIV proteins into smaller individual proteins. Okay, so as the smaller HIV proteins come together with copies of HIV RNA genetic material, we are going to have a new particle that will assemble in readiness to form a virus. So when these particles have assembled, they bud out of the cell, host cell. So stage 6 is budding. So the newly assembled virus pushes out, okay, out of the cell, creating a lot of holes. And as they push out, okay, the virus steals part of the cell's outer envelope. So this envelope is one which acts as a covering, okay, and this protein, this contains a protein and sugar combination called the HIV glycoproteins. So these HIV glycoproteins are necessary for the virus to bind, because that is where you find the other parts that the HIV needs to bind to a host cell and infect that cell. So we have been looking at HIV life cycle, and today we end on that. Please keep studying.